Uh, Hi everybody. We were going to learn something about a process that happens in the nucleus called transcription. Okay, this is transcription. This happens, we are now studying it, we are studying it in the eukaryotes. This we are studying in the eukaryote cells. That means the cells which contains a real nucleus that we are going to study. Now, what is transcription? Transcription simply means copying of the DNA information, genetic information, and then passing it on to the RNA. That is the genetic information that is required to construct or to build a polypeptide chain will be copied on a RNA molecule, that is ribonucleic acid chain, and that happens on the DNA. DNA becomes the template. From the DNA, the information is carried, and that will be carried by this RNA, and that will go to produce the proteins. This idea is already known to all of you, and this is often called the central dogma. That means in any living being, the information from the DNA, the hereditary characters will be passed on from DNA to other parts via RNA. And that process is called transcription, copying of the information contained on the DNA to carry out various processes in a cell is called transcription. Now, this happens on the DNA molecule. If that is the DNA molecule, you know the DNA molecule consists of two strands will be there, and in these two strands you will find the base pairs will be there. They will be connected by means of base pairs. This already you know in your lower classes or higher classes, so that we don't have to worry about it. Now these base pairs will be cytosine, base pairs with the guanine, and then adenine goes with the thymine. These are the base pairs here. Supposing if there is adenine here, there will be thymine here. If there is cytosine here, there will be guanine here. If there is cytosine, again guanine. Like this, it continues. So like this is continuous. That is the way the DNA is normally seen. And now let us say that this strand, we will call it the 3' end and 5' end. This will become 5' end and 3' end. Now this is the structure. Now how did we come to this one? If you say that one, that the DNA molecule, is a very long thread-like structure. It is normally called chromatin. During cell division, this becomes, this is a chromatin, and this chromatin now becomes into smaller segments called the chromosomes, and these chromosomes will contain the genes, and genes or a gene is a segment of DNA, that will carry information to produce one polypeptide chain or a protein. It will contain it can form a protein by using the polypeptide chain. Polypeptide is nothing but the long sequence of amino acids. Amino acids. So that means the gene which is present on the DNA molecule has the capacity to produce and send information to ribosomes to manufacture or to biosynthesize the polypeptide by using the correct type of amino acids. You know, there are 20 amino acids and these will be connected to form a different type of proteins. Now let us try to go a little forward on this backward idea we have given now. So now we have taken a small segment of DNA and that is taken from the gene, and this will be now transcribed into what's called mRNA. So in the transcription process, the DNA is copied to form mRNA. Now if you take this particular thing, there must be some preparations for that. It will not happen immediately like that. And scientists have recognized that there will be some important areas on the DNA molecule which are essentially used for the activity of the transcription. And these areas are called the areas for initiating the transcription. Now, we can call one of these things as an enhancer. I will tell you what they are, and this may contain accelerator and also the retarder, and this will contain the promoter. All these things are the sequences of the bases. They will do different functions. Now, the transcription actually happens in 3 important stages. Now, transcription to happen, first thing is called the initiation. That is the first step in the transcription. Second is called the elongation. And third step is called the termination. These are the 3 steps that happens in the DNA formation of the mRNA molecule. Now we have identified that the DNA contains certain special spot called the receptors to act up. Now what happens is the cell will all this will be happening inside the nucleus, that is in the eukaryotes. Nucleus contains a large number of enzymes and also proteins. So some of the things that require for the initiation will be, one of them will be the RNA polymerase. It is a enzyme plus some factors. So these together forms what's called the initiation complex. Okay, you understand that point now. So this initiation complex comes and binds this area, promoter area. That's the initiation complex. That is the first part of the formation of the mRNA. So initiation complex containing the polymerase and other things which we call the initiation complex, they will come and join here. And once that happens, we will take only this portion now. When that happens, the strands which we have taken here, this strand, now this strand opens up a little bit like that. It opens up because I am using the 2 colors here. It opens like that. So the polymerase works on this one and makes the DNA molecule to open up. And this may continue. We have taken only a short segment of it. Now, if you look at these strands, one of these strands is called the coding strand. The other one is called the template strand. So this strand. So this you should know now. So there are two things. This will be 5 prime, 3 prime. This will be 3 prime and 5 prime. Now this is the structure. Now if you want to know how it happens, now we will just write here some of the molecule, the bases guanine, let's say adenine, thymine, adenine, guanine, cytosine, thymine, adenine, cytosine, like that it continues for a long time. And we know that there is a complementary base in the other strand. So in this case, it will be for this, that will be cytosine, further that will be thymine, adenine, thymine, guanine, this will be cytosine. For cytosine that will be guanine, for thymine that will be adenine, and for adenine there will be thymine, for adenine again thymine, for this there will be guanine, like that it also continues. So this is the way we have to copy this particular segment, because we are not going to go to all the things. We are only trying to find out how this happens, the initiation and how the copying takes place. Now when these things are happening, again the DNA polymerase, that is here, DNA polymerase, this starts moving. It starts moving. As it starts moving, it comes to the first area, the first site, and that will be the site containing the cytosine. So it picks up the RNA now. Now in the RNA, what happens is cytosine is there, guanine is there, adenine is there. Instead of thymine, there will be uracil. So this is the RNA this is RNA basis, this will be DNA basis. This you should remember. So wherever you have to add thymine, then uracil should be there. Supposing we start putting that one. Supposing this the polymerase starts picking up the RNA nucleotide, it picks up the guanine and fixes it here to this opposite. And now this is thymine. For thymine, it is going to pick up the adenine. And then, it's again adenine and thymine, you are correct. So for thymine, you get the adenine. And now you got adenine here, and we have to have the thymine. So instead of thymine, you will get the uracil. Here again you got the thymine, and definitely adenine can be added here. And cytosine, guanine, cytosine, adenine is there. For that you must have the thymine. But instead of thymine, you will get the uracil and adenine, and the cytosine, like that. So that means in the RNA, this type of nuclear bases are added from the RNA, and then you will get the phosphate sugar backbone, and that becomes a now RNA chain, mRNA chain copied. This mRNA, which is formed here, is called pre-mRNA. So this pre-mRNA cannot be used for making the polypeptide chain. This has to be processed. Okay, that's what we just remembered. So now we have initiation was taken place, and this portion of the thing is called the elongation has been done, and the termination part is concerned as the copying continues in this direction. At some part of the time, the DNA sends the stop signals that may contain some base sequences, and we get the termination signal or stop signal, and then it stops. So termination takes place, and this RNA, which is produced, what's called the pre-mRNA, now it comes out of the DNA molecule, and it comes out like that. That is the pre-mRNA molecule that has been produced, and this will contain the guanine, adenine, uracil, adenine, guanine, cytosine, uracil, adenosine, guanine, like that it continues. Now this particular long chain of the RNA molecule is still requires what's called processing. It has to be processed. It is not ready for thing because the DNA molecule will contain a large number of base pairs. Some of them are redundant. They are useless. They are sometimes called nonsense codons, or sometimes they are called introns. So that has to be removed. If they are not removed, then the protein that is produced will be defective. So that for that purpose, the mRNA molecule that is produced, called the pre-mRNA molecule, will have to be processed. This process also takes place in the nucleus. To do the processing of this thing, if you just say, let's just imagine that here is one section of the mRNA that is functional, it makes, and that can be expressed. That's called exon. That can be expressed in the thing. And similarly, there may be another small structure that may be also exon. That may be one more here. That may also be exon. So there are areas where you have the functional sequences, but there may be certain spots, for example, like this, maybe one, maybe like this, two. These structures do not help in the production of the, or in the formation of the polypeptide, and these are called introns. Intron, this is also one intron. So now the splicing takes place by means of the enzyme called spisosome. Although it contains other units also. So this enzyme now comes and cuts here, splices here, and then removes this one. They will go out of the thing, and now we will be getting a structure where we have the mRNA like that. Now these are removed. Now we got here guanine is there, adenine is there, uracil is there, A is there, G is there, and this C is not there, and then we got a U, A is there, this CG is not there, and therefore this may be U, A, U, U. So we have U, A, U, U. So now this becomes definitely an mRNA. This also has to be protected because it's a very precious material. Therefore, what the cell has done it adds to this one a cap. This cap is made from the guanine compound, molecular compound, that's called the cap. And that cap again goes at the 5' end. Now this, as it is copying, this is the 5' end, and then this will be 5' end, and it goes to 3' end. Right? Now this will be 3' end. Imagine that will be 5' end, and that continues to form. So at the 5' end, 5' end, the new structures are at the 5' and that continues. Now this is 5' and 3'. Right? If you now look at that particular thing and then go at the go now, we have added the cap. Now at this end, you get a polypeptide chain. It's called A tail. We get a tail also here added. So now this becomes a complete mRNA that is ready to go out of the nucleus. And so we have now discussed the transcription that takes place in 3 stages: initiation, elongation, and termination. And the final product will be mRNA, and that mRNA goes out of nucleus, and that will go to the cytoplasm, now cytosome or the cytosol. It enters the cytoplasm, and then it attaches itself into the structures called the ribosomes. It comes to ribosomes, and in the ribosomes, lot of reactions will happen, and then a process called translation takes place. And translation will convert the coded material. Here, the codes will be made available into form a polypeptide chain. So this polypeptide chain is formed based upon the information that is received by the mRNA. So we will just stop here because the cell takes all the precautions to see that the correct information is passed on to form the proteins. Because in our body cells there will be hundreds and thousands of segments of the DNA working at the same time and producing different types of proteins which will form into different tissues, organs and functions, etc. So there should not be any mistake. For that purpose, there will be splicing, and they carefully go through that, and then only they will be sent to ribosome to make the polypeptide chain and then into protein. So next one we will study about the translation.